Okay, can everyone see the screen? Please respond. Okay, Anisa, you also switch on your video. Am I audible, Anisa?
Yes, ma'am.
All right. Okay, now see. In the previous class, meaning yesterday, I have started the biotechnology principle and processes chapter. Now, since you weren't there, so let me just quickly run it down for you. And for others, it will be sort of like a quick revision. Now, when we talk about biotechnology, it is when we integrate the technology along with the life sciences. This is done in order to make our favorable products, and you can say all of the things that will be beneficial to human beings. So when we talk about biotechnology, so it is the technology that is based on biology, especially used in the field of agriculture, in the field of food science, and in the field of medicine. These three fields, majorly. Now, there is this federation called as European Federation of Biotechnology, EFB. This has given, a proper definition as to what biotechnology is. So, according to it, the integration of natural science and organism, cells and their parts, along with their molecular analogs for products and services. Ultimately, this whole entire field or this whole entire branch of science is for the betterment of human beings so that we can derive, you can say, a lot of benefits. Now, when we talk about the principles of biotechnology, it runs on two principles basically. The first is genetic engineering and the second is bioprocess engineering. Now, when we talk about genetic engineering, see at the level of the gene, right? So it is happening at the level of the gene. So why is it done or what is this done for? It is the technique that is used to alter. Alter meaning something could be added to the like genetic material or something could be removed from the genetic material. So these are the techniques that are used to alter the chemistry of the genetic material. Now you people know that genetic material, there are of two types, basically DNA and RNA. Call it genetic material, call it nucleic acid, right? And then when we change it and introduce into the host so that when the host will divide, we will get our desired products multiplied. So this is the first principle of biotechnology. Now the second principle of biotechnology is bioprocess engineering. Now when we talk about bioprocess engineering, so this is like while you are handling with someone's or some organism's DNA, and while you're, like putting it into the host to product any, to make any kind of product or anything of that sort, at that point of time, you have to be very careful that no contamination occurs. Because if the contamination would happen, then whatever is your experiment, it will become failed. It will fail miserably. So that is why the maintenance of the sterile environment or the contamination free environment is very, very necessary. Now, when we are doing the process of this thing, uh, biotechnology or altering the chemistry of DNA or RNA, during that time, what we do is that we create a recombinant DNA that is called as rDNA. Now, these are the DNA molecules that are made in the lab, and we use certain techniques that are there in the biotechnology. Thereby, you take the gene of interest, put it into another organism part like vector, and then you introduce it into the host. So what you've made is a recombinant DNA. And this whole entire chapter is based on recombinant DNA technology. This much is clear, students? Any doubts? So this was the introductory part to the chapter. Now there are a lot of, you can say, raw materials or a lot of steps that go behind into making of the recombinant DNA. So first of all of you, if you remember in the previous class, I had told you about cloning. Now when we talk about cloning, so it is a process of making identical copies, that too many identical copies, right? Anything, if it gets cloned, then that means multiple copies of that thing is being made. Now talking about the basic steps in the process of cloning. So the first is the construction of the recombinant DNA, rDNA, right? How? You take the gene of interest, okay? You take the gene of interest, and when you're going to add it into a carrier molecule, that is the vector, ultimately what you have is the recombinant DNA. Now this recombinant DNA, it has to be sent into the host cell in order to, you can say, get the desired products. So now the recombinant DNA gets transported into the host cell, and when the host cell will divide, like for example, bacteria is the host cell. So when the bacteria will divide, then the recombinant DNA will divide. Okay, and then basically it will go into the further generation. Now moving forward, like we talked about, the first recombinant DNA, it was made by two people, that were Stanley Cohen and Herbert Boyer, and this was done in the year 1972. Now what they did is that they took out the strain, or you can say they used Salmonella typhi. Which disease does Salmonella typhi cause, a student? Salmonella typhi is the causative organism of, tell me. Whoever has to speak, you speak. Unmute and speak. Quickly. And students, why are you always quiet? I don't understand. Salmonella typhi is the causative agent of?
Typhoid.
Typhoid. Obviously, it's there in the name. Why are you people so confused? So what they did whilst you can say discovering or making the first recombinant DNA molecule is that they took the plasmid of Salmonella typhi and they linked a gene with it, and thereby the recombinant DNA was made, and this was done in the year 1972. Students, do you have any doubts understanding anything that I've said up until now? Hmm?
No, ma'am.
Okay. Now further, there are three basic steps when you try to genetically modify any kind of organism. The first step is that you see the gene of interest, right? Whatever is the interest of the gene or the part of the DNA that you people require, first you identify that. Secondly, after the identification of it, you put it into the host. Now, you cannot directly put it into the host. Rather, you will send it into a carrier, and then the carrier will take it into the host. And lastly, you have to maintain it in the host so that you can get the desired product. So these are the three basic steps that are employed during the process of the genetic modification, the production of GMO. What does GMO stands for? Genetically modified organisms. Clear? Is it clear to everyone up until now? Now see, during the process of DNA technology, like recombinant DNA technology, you require certain raw materials, you require certain, you can say, tools. Now, when we talk about the tools, what are they? First, you need, that you will be needing the DNA manipulative enzymes, meaning these are the enzymes with the help of which you can manipulate the DNA. You can manipulate how the DNA would be altered. Okay, you cut it, you join it, all of that. So for that, you have restriction enzymes, you have polymerase enzyme, and then there is ligase enzyme. The second thing that you require are vectors. Now, vectors are just like your carriers and they will be carrying the gene from one area to another into the host cell. And obviously, all in all, you're going to put everything into the host, so you require a host organism. So these are the basic requirements. So in this chapter, you're going to study more about the tools and how you are going to use the tools that are there or that are used during the process of recombinant DNA. Now, when we talk about restriction enzymes, they are in very simple terms called as molecular scissors. Just like how we have normal scissors. And what do we use the scissors for, students? Tell me. This also you don't know. Scissors, what do you use scissors for? Nothing, ma'am. We don't know how to use scissors. Eh? What do you use the scissors for? Answer. Cutting, right? Cutting thing. So when we talk about molecular scissors, so these are the ones that are used to cut the molecular level things. Okay. They are also called as the chemical knives. Now, what happens is that the restriction enzymes, they cut the DNA into certain fragments. So however many fragments you would want, it would cut it into those fragments. Now, the restriction enzyme, they belong to the larger class of enzyme that is nucleases. Nucleases is like the big class of enzyme under which restriction enzyme comes. Now, nucleases are the DNA cutters. Now, they are of two types, depending upon where are they going to make a cut. Okay, if they are going to make a cut in the middle, you call it as endonuclease. And when they are going to cut at the end, then you call it as exonuclease. So, in the exonuclease, it removes the nucleotide from the ends of the DNA molecule. Okay, whereas when we talk about the endonuclease, so what they do is that they're going to break the internal phosphodiester bond internally. Like for example, if this is a DNA molecule, okay, if we are talking about the function of exonuclease, so it is going to cut from here and it is going to cut from here at the ends of the DNA. Whereas in the second case, when we talk about the endonuclease, it's going to cut in the middle. Okay, so this is the major difference, that the endonuclease cuts in the middle, right in the center of the DNA, and it breaks the phosphodiester bond. The exonuclease, it removes the nucleotide from the ends of the DNA. It cuts or cleaves at the end of DNA molecule. Now, the first restriction endonuclease that was isolated, it was HIND2. And how does it function? It always cuts the DNA at a particular point. Now, remember students, the particular point where the DNA will be cutting, every DNA has a specific point where it will be cut by the restriction enzyme. So it recognizes a sequence of 6 base pairs. Like once it is done recognizing, like for example, ATGC, ATGC, once it's done recognizing the 6th one, then it will cut over there. Now there are over 900 restriction enzymes that were isolated, and these enzymes are actually, you should know that where are these enzymes coming from? They are produced naturally inside the bacteria. Okay, so we extract it out of them in order to use it. So there are over 900 restriction enzymes that are present, and they are isolated from over 230 strains of bacteria, each of which are like going to recognize different sequences. Always remember, like the restriction enzymes, they're going to inspect the length of DNA. Okay, they inspect the length and they find out which is the sequence that suits them, and then only they make a cut. Okay, any doubts? Now the question comes along, why are the bacteria making these enzymes? Why? What is the need? So it's like it's their defense mechanism, because you know that bacteriophages are those viruses that infect the bacteria. So to protect them from those viral attacks, they produce the restriction enzyme. Now let us see some of the examples of the restriction enzyme. So there is BAM-H1, there is EcoR1, then there's HIND3, PST1, SAL1, SMA1. These are the different names of the restriction enzyme. Each of these names you should be familiar with. Now see, all of these like names of the enzymes that you're seeing, it's just not very random. It has a particular name associated with it. Okay. So when we talk about the naming of restriction enzyme, so this is how you name it. The first alphabet in the name is the name of the genus of that bacteria from where it is taken. The second and the third is from the species of the bacteria from where it is taken. The third letter is the strain, whether it is a rough strain or smooth strain like that. And then the Roman numeral, okay, Roman numeral, it indicates the order of isolation, whether it was, taken out first, a second, a third, a fourth, like that. So always remember this nomenclature standard. Okay, so this particular Ecor R1, this one is extracted from Escherichia coli. That is the most common bacteria there is. Now let us see how does the restriction enzyme work. Up until now, is everything clear, students? Is there any part you people did not understand?
No, ma'am.
Oh, very good. Now, moving forward, talking about the working, how these work. So, the restriction enzyme, they're going to recognize a specific sequence in the DNA. And then, like I told you, then it is going to make the cut. So, a palindrome nucleotide sequence in the DNA, like palindromes are like these words, like how they start from the same alphabet, they end at the same alphabet. These are palindromes, like mom, M-O-M, mom. If you read it from the backwards also, it's the same thing. Malayalam, if you read it from the back also, it's the same. So palindromic sequence they inspect and then only they cut. So each restriction endonuclease, it functions by inspecting the length of the DNA, and once it finds the recognition sequence, then it will bind it and then cut from there. That's how it happens. Okay. So now when the enzymes are cutting, for example, if you are cutting any paper, so like you cut in the middle, so you have two parts of the paper. And then if you cut it tilted, the tilted parts will be there. If you cut it straight, the straight parts will be there. Same way, when the DNA is cut, so it creates two types of ends. The first one is called as the sticky end, and the second one is called as the blunt end. So when the restriction enzyme is going to cut the DNA little away from the center, then it is going to make sticky ends that are also called as staggered ends. But when it cuts right in the center, not away from the center, right in the center, then it is going to generate blunt ends. Now, always they remember this as a rule that the vector, like once you cut the DNA, then you will load it onto a vector. So always remember that the vector and the DNA, they must be cut with the same restriction enzyme or else they will not be joined ever. So, for example, if you are using EcoR1 to cut the DNA, then you have to use EcoR1 to cut the vector as well. This is the rule because either it will not join. And after that, after you're done, like how will you join the DNA, cut DNA and the vector? So you use the DNA ligase enzyme, which is called as the molecular glue. So what it does is that it joins the fragments of DNA and how it forms the phosphodiester bond. Is it clear, students, up until now? Have you understood how the restriction enzyme works and how the glue works? Okay. All right, now you people note down till here, then we'll see the function of DNA polymerase. So, Anisa, since you missed the yesterday's class, you start from here onwards, okay?
Yes, ma'am.
Done, students? Shall I scroll down? Also, once you people are done, unmute your mic and then tell me, ma'am, I'm done. Okay?
Ma'am, done. Done, ma'am.
Everyone's done till here, students? Yes, students, everyone's done?
Yes, ma'am.
Done, ma'am. Done, ma'am.
Everyone's done till here? Everyone's done? Done? Everyone's done? Yes or no?
Yes, ma'am.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
All right, everyone's done till here. Now see students, furthermore, let's talk about DNA polymerase. Now all of you have seen how DNA polymerase works, right? If it is a DNA dependent DNA polymerase, obviously it will synthesize the DNA depending upon the template DNA that is provided, right? So it synthesizes a new strand of DNA. People have seen it in the replication process. Now, the next thing is the cloning vector. Now you like I told you that how vectors are important as far as the transmission of the or the carrier of the gene of interest is concerned, right? So these are the carriers, and you use the cloning vector in order to send the gene of interest into the host organism. Furthermore, when we talk about the features that must be there in a vector, what are they? The first thing that every vector must have is the origin of replication. Now, this is the point in that particular vector from where the replication process will begin. So, if let's just say the gene of interest is linked at the origin of replication, then it will replicate along with the replication of the host. Now, the process of cloning or making multiple copies, first of all, like in this way only, like you take the gene of interest, ligate it onto that part which has the origin of replication, and thereby create multiple copies. Now, the process of cloning or making multiple copies is done exactly like that. The second thing that a vector must have is selectable marker. Now, selectable marker are the ones, like for example, there is a vector that has antibiotic resistance. Now you put a foreign gene and they will lose the antibiotic resistance. Now when you will grow, like if you, for example, if you're growing them on ampicillin, they will not be able to grow if the ampicillin resistance gene has been removed from them. So now when you when they are not growing, then you can confirm that, okay, these are the ones whose DNA have been modified, whereas the others are the ones which do not have the modified DNA. So this selectable marker is very, very important in order to for you to distinguish which one is a transformant and which one is not a transformant. Now, there are many genes in prokaryotes that code for antibiotic resistance, right? Such as ampicillin gene is there, chloramphenicol gene is there, tetracycline, kanamycin, all of these are the different antibiotics that are there, and like these are the resistance site. Like meaning this particular bacteria which will have the resistance site of all of these, it will be resistant to the activity of these antibiotics, as a result of which they will not be killed. But now when you put your you can say DNA of interest or the gene of interest into these vectors, if you put it at the site of ampicillin, it will lose the resistance. If you put it at the site of tetracycline, it will lose that resistance. So that will help you to identify which of the vector is transformed and which one is not. Now, this is the method wherein you do constant plating, right? You put it on this medium and then you add the antibiotic, then you put it on another medium, then you add another antibiotic. So in this way, it is going to get very cumbersome, very tiresome also. You have to do plating here and there. So now if it produced some sort of color, it would have been easy to visualize it and to identify it. So some enzymes like beta galactosidase that is coded by the LAC-Z gene, it can also be used for the selection purpose. Now what it does, like once it produces the color. Okay, so it produces color. Now, if you put the gene, it will not produce the color. So once that are producing blue color, you will understand that this has not been transformed. But the ones that produce the white color, then you will understand that this is a transform. So this selectable marker is clear to everyone. Hmm? Is it clear or not, students? And then there is the cloning site. So this is the point where you will clone the piece of DNA. And then the size of the vector should be small. It should be having a very high copy number. Any doubts up until now? Yes or no? Have you people understand this much or not?
Yes, ma'am.
Okay, no doubt.
Done, ma'am. Done, ma'am.
Okay, done, students?
Yes, ma'am.
Okay, so for today we'll keep it till here only, and everyone will study till here. Whatever notes you have, you people will come prepared with it, because before teaching you the next topic, I will ask you people questions. Okay, so everyone must come prepared with this. Okay, students?
Yes,
Yes.
ma'am.
All right. Okay, then I'll see you all in the next class.
Ma'am, I missed
Yes.
yesterday's class, so will it be compensated?
What?
Ma'am, I missed yesterday's bio class, so will it be compensated?
I did teach you from the beginning today.
Okay, ma'am.
Yeah,
Okay.
the notes that are left, you take screenshots of those notes, the ones that were in the beginning, so that you would also be prepared with it. Okay, so you start taking screenshot. Wait,
Yes, ma'am.
weren't you paying attention in the class?
I was, ma'am, but I got disconnected in between many a times.
Okay, you take a screenshot. Tell me once you're done.
Done, ma'am. Done, ma'am. Done, ma'am.
Today, just for you only, I began from the starting. Like I had, like completed quite a lot, but since you were not there, so I did it again for you in today's class. Okay.
Yes, ma'am.
Okay. Now take a screenshot.
Done, ma'am. Done, ma'am.
And you're taking the screenshot, so you complete your notes also, right? Write it down in your
Yes,
notebook.
ma'am. Yeah. Yes, ma'am.
Done.
Done, ma'am.
Also, dear, when will you get your this thing fixed? Camera.
Yes, ma'am. When I turn on my camera, like your presentation gets slow and your camera also gets stuck. But when I put it off, like everything goes normal.
Okay. Done.
Yes, ma'am.
And then from here onwards, you wrote, right?
Yes, ma'am. I wrote.
All right. Okay. Then before the next class, you come prepared. I will ask questions. Okay. You study it nicely throughout the week, and then we'll meet on the weekend. Okay.
Yes, ma'am. And ma'am, what